Today we'll start with the hallmarks of cancer. So before entering into the hallmarks of cancer, so we have to look after some of the few terminologies we must understand, right? So like anaplasia, dysplasia, hyperplexia, and hypertrophy. So what are all these terms represent is that anaplasia, which means the lack of differentiation. So what is differentiation is that the normal cell is keep on dividing, right? So when in the case it is getting differentiated, so once they have the structural morphology, structural features must be developed, right? So once it is getting differentiated, so they will be obtaining the structural features in it. So those structural features is uh, um, just in the case they are lacking those features, it means the lack of differentiation is there. That term is called as the anaplasia. Whereas in case of the dysplasia, so loss in the uniformity of the individual cell. So or just how we will be representing the lack of differentiation. So dysplasia is that structural features, for example, the structural compartment features like formation of cytoplasm and nucleus, everything would be arranged in such a manner that the normal cell must perform in a good way. So that uniformity in the or a structural abnormality will be happening. That is called as the dysplasia, right? And then third is the hyperplasia. So hyperplasia means that the cell numbers is increases, keep on increasing. For example, if a single cell divide by means of one cycle means it will be the one becomes 2, right? Whereas the cell number is keep on increasing in an irregular manner. That is called as a hyperplasia. Whereas hypertrophy, so the hypertrophy is the increase in the cell size. So even the cell numbers are increasing means that cell size is also abnormally increases. That is called as a hypertrophy. So anaplasia, dysplasia, and then hyperplasia and hypertrophy. So these four terminologies we must keep it in mind. So already we are aware of the metastasis also. So what is the metastasis? So the normal cell is getting mutated. They will be converted into the cancerous cell. So that conversion is called as a neoplastic transformation. You already know, right? So once the normal cell is converted into the cancerous cell, they will be the cancerous cells will keep on dividing in a site of origin. So in which site the cancer cell is starting to develop, that site they will be developing, right? So that is called as a primary tumor, site of the primary tumor. So from the primary tumor they will be migrating to the local tissue or the distant tissues. So they will be keep on migrating. So once they after migration it can be developed into the secondary tumor, right? So that is called as the metastasis. So already we know that it is called as metastasis. And then so if the cell divides, so normal cell if it divides, so this is how it divides. Whereas once they divide, they will be arranged in a layer form. If it is arranged in a layer in a proper manner, regular manner, the layer is getting arranged, right? So it is called as a regulated growth. So if any cell, a particular cell is getting mutated, means that in so that if it gets mutated means how does it going to escape? Normally it will go for apoptosis, right? So if the apoptosis happens, those cell will be cleared over here. So the gap is generated over the layer, right? So once the layer is getting disturbed, so what does it would result? It will be resulting in an unregulated manner. So the layer is regulated over here in the unregulated manner. This is how the unregulated manner, like it is not in the proper proximity. This is how the cancer cell in an unregulated manner. This is how the layer formation will be happening, right? Then, so how the normal cell and cancer cell is differentiated. So the normal cell, the first is that they will be having the largest cytoplasmic matrix. Meanwhile, the nucleus is single and nucleolus is there and the chromatin is very fine. In case of the cancer cell, cytoplasm is very smaller and also nucleus are there having the multiple nucleus and nucleolus is also multiple and the chromatin is very coarse. So this is how the normal cell and cancer cell would be, right? So and then moving to the hallmarks of the cancer cells, these are all some of the hallmarks, which means that the increased nuclear cytoplasmic ratio. So the increase as nuclear cytoplasmic ratio, when there will be the increase nuclear cytoplasmic ratio, which means the surface to volume ratio is increasing, which means that cell size is bigger. Cell size is bigger, that is called a hypertrophy, right? So cell size will be bigger. So if the cell size is big, means their nuclear and cytoplasmic ratio is also obviously increasing. And also we know that they will be having then multiple nucleus and multiple nucleolus. So the multiple nucleus and multiple nucleolus would result in the formation of the more number of the genetic content and also more mitotic figures will be there, right? So and then chromosomal number is also varies, and the loss of the contact inhibition. So the contact inhibition is that so once the cell is just started dividing, so they will be keep on arranging in a layer, right? So that each and every cell is contacted, so that once it is getting contacted by another cell, there itself the cell division will be stopped. So once there is no even though if they get contacted, there is no inhibition, they keep on dividing. So the normal cell, if it is getting contacted, the cell division stops, whereas here the cancer cell is contacted, they will be keep on dividing, which means that no inhibition over there by means of the contact. And loss of the anchorage dependent, which means that they need some platform to divide, they need some environment to divide, right? So consider we are growing the culture in the petri plates. So the petri plates is an anchor over there. There they will be having some space to divide, to undergo the division in a certain boundary. Whereas in case of the it is called as anchorage dependence. Whereas in the cancer cells there will be no restrictions. They are not at all need any micro environment or any boundary or any supporting base to develop, to divide. So they are not at all depend on anything to develop, to divide. That is the thing. And then the secretion of the growth factors. So secretion, they will be, these cancer cells possess a specialized growth factor, so that it will be meant for the irregular division also. And then the expression of the growth factor receptors. So how the growth factors is signaling, and they will be elevating the growth signals. Meanwhile, the growth factors receptors is also essential for the that signaling mechanism, which is also expressed in a larger manner, and then this paved a way for the growth of the cancer cell more effectively. Consider, so this is our bloodstream having a lymphatic system, and lymphatic blood vessel is there, it will be shown here. So this would be a normal cell. So if the normal cell is mutated, it undergo hyperplasia, right? So once a hyperplasia, which means that the cell numbers are increased in size, whereas next it will be converted into this is the normal cell. Once it is getting undergoing hyperplasia, it becomes cancerous cell. So once it is getting cancerous, they will be converted as a primary tumor. A primary tumor at the site of origin, only at a particular site it will be it will be considered as tumor. If it undergo metastasis, sorry, if it what does it do? They have to cross the blood vessel to undergo the metastasis, right? So this is the initial to convert into the secondary tumor. So this is how it follows the path. And then development of the malignant tumors. So malignant tumors are easily developed in a highly proliferating cells. So first initially the cancer definition itself, they are very much prevalent in the proliferation. Cell proliferation and differentiation is very prevalent. We already know about that. So the most cancers is on highly proliferating cells. It occurs in the, it can easily accustomed over there, right? Like epithelial cells and stem cells. So what is the property of the epithelial cells and stem cells? They can be converted into any type of cell, which means that they can be called as a pluripotent cell, right? So apart from the uh, apart from the cancers are very much suited for the proliferating cells. They can easily survive over there. Meanwhile, the environment like chemical environment and then viruses infection and so many environmental factors is also contributing the cancer cells to develop very easily, right? Coming to the hallmarks of cancer, totally 7 hallmarks are there. One of first is that self-sufficiency in the growth signals. So what is it called as a growth signals? So for the normal cell to perform, signal is very important. So what is the purpose of signal? To move on into the normally if we are standing in the traffic, so we need one signal to cross the road or to cross the path, right? So meanwhile, for any progress to happen, signal is mandatory, right? Signal is having greater role, right? So for anything to happen, meanwhile so this growth signals are very sufficient for the cancer cells are divide. They will be having an very pakka growth signals. Meanwhile, they are very insensitive to the growth inhibitory signals. So each and every action have an opposite reaction, right? So even though growth supporting factors are there, growth enhancers, promoters, whatever promoters, enhancers, so many things will be studying the cell biology and molecular biology, right? So meanwhile, inhibitors is also there. Inhibitors role is also there. So they are very insensitive to that inhibitors, so that nothing would stop them to grow from dividing. And then aviation of the apoptosis is the third thing. So what is aviation of apoptosis? So normally when the apoptosis will happen, if the cell undergo damage, they will be trying to repair it themselves. If the repairing mechanism doesn't work it out, so what does it happen? Normally it undergoes the suicide, right? If it undergoes suicide, it will lead to apoptosis. If it doesn't undergo suicide, means what it would do, it must be murdered, right? By means of treatment, we are murdering those cancer cells. But normal cell can undergo apoptosis, whereas in case of the cancer cell, this apoptosis is also escaped by the cancer cell. The cancer cells keep on proliferating, they don't undergo apoptosis like a normal cell. And then defects in the DNA repair. So we just what we see for the apoptosis, DNA repairing mechanism is most important things. See, if that checkpoint fails, if the DNA repairing mechanism fails, no, that is the privilege for the cancer cells to develop very, very easily, right? And then limitless replicative potential. We already know that one. By means of replication only the cell division happens, so that that is not limited at all. In case So if it is limited means it will be regulated. So there is not at all, they are not having any limit at all. And then sustained angiogenesis, right? And then last is the invasion and metastasis, right? So these are all the hallmarks of the cancer, and we'll stop here.